Hello friends, in this video we will discuss lipid metabolism. This is the overview of lipid metabolism. In this that triacylglycerol in the storage form or structural lipids, all these are undergoes beta oxidation where the APDH2 and NADH molecules are produced and NADPH or ATP is used to convert that fatty acid into acetyl-CoA, and that acetyl-CoA undergoes TCA cycle or citric acid cycle. Then citric acid cycle produces NADH, APDH2, GTP molecule which undergoes oxidative phosphorylation for the generation of the energy. Fatty acid oxidation, it may be defined as the oxidation of fatty acid on the beta carbon atom results in a sequential removal of the two carbon fragments, that is acetyl-CoA. In this fatty acid oxidation, three stages are there. First stage, that is the activation of fatty acids occurring in the cytosol. Then second stage, that is the transfer of fatty acid into mitochondria. Third stage, that is beta oxidation, takes place in mitochondrial matrix. Fatty acids are oxidized by most of the tissue in the body. However, brain, erythrocytes, adrenal medulla cannot utilize fatty acid for the energy requirements. Here is the first step, that is the activation of the fatty acid to acyl-CoA. Fatty acid is converted into acyl adenylate with the help of enzyme, that is the thiokinase, where the ATP is used, and that ATP after using ATP, inorganic pyrophosphate is produced, which is converted into inorganic phosphate, that is with the help of enzyme, that is the pyrophosphatase. That acyl adenylate is converted into acyl-CoA. That acyl-CoA is synthesized with the help of coenzyme A, or there is a formation of the AMP. Next step, that is the transport of the acyl-CoA. As such, fatty acid is not able to transfer, is not permeable to fatty acid. There must be conversion for fatty acid into acyl-CoA. So the inner mitochondrial membrane is impermeable to fatty acid. A specialized carnitine shuttle is used to transport activated fatty acid from the cytosol to mitochondria, and this process takes place in four steps. First step, that is the acyl group of acyl-CoA is transferred to carnitine, and that reaction is catalyzed by carnitine acyl transferase. It is present on the outer surface of inner mitochondrial matrix membrane. Then the second step, that is the acyl carnitine, is then transported across the membrane to mitochondrial matrix by a specific carrier protein. In the third step, that carnitine acyl transferase 2 is present in the inner surface of inner mitochondrial membrane, convert that acyl carnitine to acyl-CoA, and in the fourth step, that carnitine release returns to cytosol for reuse. This is the diagrammatic presentation how the carnitine transport system operates, transfer that acyl-CoA from cytosol to mitochondrial matrix with the help of carnitine shuttle. Then next is, that is the beta oxidation. Actual beta oxidation, we were liberating a 2 carbon unit acetyl-CoA occurs in a sequential reaction, that is oxidation, hydration, oxidation, and then cleavage. On the right side there is a reaction flow chart, that is how that long chain fatty acid is converted into fatty acyl-CoA. That is the first step, activation of that fatty acid with the help of enzyme, that is the thiokinase. Fatty acyl-CoA then undergoes dehydrogenation reaction with the help of enzyme, that is the acyl-CoA dehydrogenase. This is alpha, this is beta, where that particular removal of that hydrogen takes place, formation of the double bond, and in this reaction APD is converted to APDH2. There is a formation of trans-enoyl coenzyme A. That compound undergoes hydration reaction with the help of enzyme, that is enoyl CoA hydratase. There is a hydration reaction, it means addition of the water breakdown of double bond. So on beta position OH group is attached, that compound is beta hydroxyacyl CoA. This beta hydroxyacyl CoA undergoes dehydrogenation reaction with the help of enzyme, that is the beta hydroxyacyl CoA dehydrogenase. In this reaction NAD is converted into NADH. Dehydrogenation on beta, that is the formation of the double bond, formation of carbonyl group, formation of that beta ketoacyl CoA, and that beta ketoacyl CoA undergoes cleavage reaction with the help of enzyme, that is the acyl CoA acetyl transferase, where the coenzyme A is used. Formation of fatty acyl CoA 14 carbon and acetyl CoA 2 carbon. This acetyl CoA further goes for Krebs cycle, and there is a generation of the energy from the fatty acid molecule, while the 14 carbon second product 14 carbon again undergoes beta oxidation in the same way. In this way, that is oxidation, hydration, oxidation, cleavage, all these four stages produces acetyl CoA. So 14 carbon again convert into acetyl CoA, then 12 carbon convert into acetyl CoA, 10 carbon. In this way, from 16 carbon containing long chain fatty acid produces total 8 acetyl CoA molecules. Thank you.